Okay, gene prediction. Finding the genes in the genomes. So we have sequenced some genome, right? And then we have done some assembly to produce some contigs. We did this a couple of modules ago. And now we want to do what we call annotation of these contigs, right? And that involves finding the genes. Where are the genes in these contigs or genomic sequences? And what kind of genes are they? Actually, that's the annotation itself. But first of all, we need to know where are the genes. And then there are sort of two distinct ways of approaching this. The extrinsic approach or reference-based means that you actually compare to some database. You have a collection of already known genes, and you see if you can find something similar here. The intrinsic approach, or de novo, means that you go hunting for new genes, you could say. Because in this over here, you can only find what you have discovered before. Here you can discover something new, right? And of course, when we started sequencing genomes, this approach was impossible because there was no databases, right? So this is the one we have sort of relied on from the beginning. And and this recognizes the genes based on some rules or patterns without using directly the external data. We have to say that it also somehow uses ideas, at least from previous seen genes. But if you think back to the very first genomes that were sequenced, there were actually very few ideas to lean upon. But now, at least nowadays, we have something more. But the de novo or intrinsic approach to gene finding is still very much in use. We will see why. If we consider the prokaryotic genes, what are we talking about? First of all, the ribosomal RNA genes. They are always there. It's the 5S, 16S, and 23S gene. They are usually located next to each other in an operon. And all prokaryote genomes have them, from 1 to up to 15, 16 copies, actually. And these are typically some of the repeated regions. We have talked about repeated regions, right? When we talked about sequence assembly, the problems we face when we have repeated regions, these are very classical repeated regions. This operon here, if you have 15, 16 copies of this, and this is quite long, right? It's like 3,000 bases at least. And if you have the same genes 15 times in a genome, you have a lot of problems assembling this piece. So they actually very often come out as a separate contig in our assembly. That's difficult to attach to the rest. The tRNA genes, they are also in there, and they are coding for the amino acids. And just like the rRNA genes, the tRNA genes are often very conserved, right? So if you have seen them in one organism, you will recognize them in another organism because they are quite the same. And there are also some other non-coding RNA genes and leader peptides and so on. And these are typically genes we find by this extrinsic approach, right? Because they are conserved. If you have seen them in one organism, you will recognize them in another one because they are quite similar. However, the protein coding genes, they are different. First of all, there are many, many more of them. This is the vast majority of the genes. These are only a few ones, right? It's like 3 of this one, 20 here, and so on. But these are thousands. And there's usually around 1,000 genes per megabase per genomic sequence. So if an average bacteria is like 3 million base pair, it has around 3,000 genes. All these protein coding genes, or coding genes as we call them in bacteria, they are made up of one contiguous open reading frame. And we will see that in the next slide, open reading frame, ORFs. And they have no introns like we are used to in eukaryotes. There are a few exceptions to this even in bacteria. There are some cases of introns as well, but in the basic rule is no introns. So that makes them a little bit easier to recognize in bacteria compared to in eukaryotes. These type of genes we look for by this intrinsic approach simply because they are not that conserved. You will not find, if you have sort of a new organism, you will not find all the coding genes by just looking up what you know from all other organisms, because coding genes can evolve, they evolve pretty fast and they can be quite different from one organism to another. But this is the ones we are going to focus on now, because these ones up here, they are sort of easy to find, right? These are the difficult ones, and these are also the majority. So it makes it so much more important. Open reading frames or ORFs. What is this? Well, let's have a piece of a genome here. Double-stranded DNA, right? Positive direction here, positive direction there. This is how it looks like. Then all open reading frames ends with a stop codon. All genes, coding genes, who are also open reading frames, end with a stop codon. And these are the possible stop codon in bacteria: TGA, TAG, or TAA. And the nice thing about the stop codon is that they have no other sort of meaning. They only mean stop of an open reading frame if they are in the correct reading frame. There is one exception I like to mention here, because there are one branch, you could say, of the tree of life where this stop codon here doesn't mean stop. It's used as an ordinary codon, coding for one of the amino acids, tryptophan. So if you work with mycoplasmas, and we actually do that here at KBM, and mycoplasma is an important bacteria. So if you happen to come across a project where you study mycoplasma, please remember the stop codon in mycoplasma is only these two, not this one, because this one means tryptophan. But in all the others, there are 3 stop codons. An open reading frame also starts with a start codon. And here are the possible starts codon in prokaryotes: ATG, GTG, or TTG. So there are 3 possible stops and 3 possible starts. These are not, there are a few exceptions to this, but they are very few and we can safely ignore them here now. This one is the most common one. It's like 80% of the genes starts with ATG. 15% of this one and only 5% of this one. So you can say 80, 15, 5. That's roughly the distribution here. The problem with the start codon is that they also occur inside genes as ordinary codons. So you never know really if when you see ATG or any of these 3, is this start of an ORF or is it just inside an ORF? You never really know. These ones we know this is the end. You cannot go beyond a stop codon, but you can go beyond the start codon. We will see that shortly now. So how do we actually find ORFs in the genome up here? Well, we start by looking for stop. And here I found a stop codon in this genomic sequence, TGA, right? And now I sort of fix the reading frame, as we say, because I now want to retrieve the ORF that end here, that has this one as a stop codon, right? And how do I retrieve those ORFs? Well, I start at the stop and then I move upstream in triplets, right? 3 and 3 bases like this. In this reading frame, I collect new ORFs, and here I found new codons, and here I found a start codon. Can you see that? GTG. In the same reading frame as the stop. But it has to be triplets, right? All the time. So this is an ORF. What we see here is an ORF. Starting here, ending down here. But the point is now that this might not actually be the start of an ORF. This could just be in the middle of an ORF. We need to go further upstream here and looking. And here we found another possible start. Right? So this is also an ORF, starting here and ending here. This is also an ORF, and the first one is just a part of this longer one, right? And G doesn't mean a start anymore. It's just an ordinary codon inside. And so on. We have to just move upstream and upstream and upstream. And here we found yet another start codon, a third ORF that sort of covers both the two first. And then we find the next stop codon inside the same reading frame. Do you see that? This is a stop, TAA, right? And now we don't need to go further upstream here. No need to go further upstream, because as long as we are in this reading frame, the ORF that ends here cannot start further upstream than here, because then it would end here instead, right? Since this is a stop. So for this stop codon here, we found now 3 possible ORFs. This one, or this one, or this one. 3 possible ORFs. The one which is most upstream of this, we call the longest ORF, or just LORF. This is my short for longest ORF, the LORF. And a LORF is simply the longest ORF for a given stop codon, right? So you have to nail down a stop codon first, then collect upstream of that all the possible ORFs, and the one which is sort of most upstream is the longest ORF, or the LORF. And this is often what we are interested in, the longest ORF, because if we have a real coding gene here now, we don't know if this is the correct start, or this is the correct start, or this is the correct start. But anyway, we always start with the LORF and investigate that first, right? Because it might be that the whole thing is rubbish, right? And then we don't care which is the correct start, because there is no sort of a start, right? So that's why we are interested in these LORFs. That's sort of the first place we start evaluating if this is a gene. We should, before we move on now, we should also indicate that this is not the only ORFs we can find here, right? Because this was for one fixed stop codon with the given reading frame. But there are also other stops here. For instance, here. Can you see that? The TAA here is also a potential stop codon, right? But in a different reading frame. And now we have to trace again triplets upstream from this one, right? Here and then here and so on, right? And you can see this will not be the same triplets because we are now in a different reading frame. And so on. So we can go hunting for more ORFs here now. Or we can, of course, also go looking at the other strand, really, right? Because this was both on the positive strand. What about the negative? Genes can be there as well. And then we have to reverse complement everything, right? And how does the stop codon look like then? Well, I think here we have one. Because if you take the complement here of this one and read it backwards, right? Because the arrow is going the other way. This is TGA, right? So here is actually a stop, and then we have to go looking for the upstream triplets here as well. Exactly the same to find the LORF and all the other potential start codons inside, right? So what this shows is that even in a fairly short piece of genomic sequence like this, you find many ORFs. You find many ORFs. Stop codons will appear all the time, you could say, in different reading frames. There are 3 reading frames on each strand, right? 6 reading frames. And these ORFs can overlap and be nested inside each other. So there are many, many, many ORFs. And actually there are roughly 100 times as many ORFs in a genome as there are real coding genes. 100 times. So in an E. coli genome, you have like 5,000 genes, but you have 500,000 ORFs. So finding the coding genes, we can think of it like this. Find all the ORFs and then classify them as either real coding gene or some rubbish ORF, right? Which doesn't mean anything. So you can think of it like this. You have the ORF sequence here from start codon to stop codon. Is this a real coding gene? Okay, then you put it into this bucket. Is it rubbish? You throw it into this bucket. And this bucket is so much bigger, right? It's 100 times bigger than this one. So it's a classification problem where you sort of look for a needle in a haystack. What kind of information do we use then to classify these ORFs? What do we look for when we have collected the ORFs? Well, there are 4 pieces of information I want to mention here. First of all, the length of the ORF itself. Well, that's pretty easy. Then the content of the ORF, which is a little bit more tricky, we will see. And then the upstream sequence. What is upstream of the start codon here? Is there some signal here we can look for? And also the overlap with the other genes. That's also a piece of information. So first, the ORF length. Why is this important? It's important because real coding genes tend to be longer than any random ORF. So random ORFs, they follow a geometrical distribution. And what is this? What is a geometrical distribution? Well, we have talked about previously the binomial distribution and coin flipping, right? This is also coin flipping, but in the binomial distribution, we first fixed the number of coin flips. We decided how many times we want to flip the coin. And then we flipped the coin and we counted how many successes or heads, right? And there is a certain probability of head and this 1 minus that probability for the other outcome. There's always 2 outcomes, right? Either success or failure, heads or tails. This is the same, but instead of fixing the number of coin flips, you just start flipping the coin and then you count how many coin flips do I need before I see the first head or success. And this is exactly what we do when we sort of think about an ORF. You start at the start codon, right? And then you sort of flip a coin. Is the next codon a stop? It's a certain probability of seeing a stop, right? Rather small, I suppose, but not 0. And if it's a stop, then you are at the end of the ORF, and the length is 1, right? But if it's not a stop, then you add 1 codon and you flip your coin again. Is the next codon a stop? And if it's not, then you add another codon and you flip the coin again, right? So the length of the ORF is like how many codons can I lay down before I see my first stop, right? And that's why it appears to be like a geometrical distribution. However, this depends on the GC content of the genome. Let me show you here. Here I have taken 2 different bacteria, Bacillus cereus, who has a low GC content, and Pseudomonas aeruginosa with a high GC content. And look at how the length distribution varies between these two. So here we have ORF length in number of codons along the x-axis, and the black curve is the exact theoretical geometric distribution for the given GC content here and GC content here. And why is it different? Why does it depend on GC content? Because remember what the stop codons are. They are either TAA, TGA, or TAG. There's a lot of T and A in there, right? And only G in two of them, right? So if you have a genome with little GC, which means many T's and A's, there will be more stop codons. So the length from any random position to the next stop is not very long, right? Most ORFs come out short because the density of stop codons is higher. You never travel very far along a genome before you see the next stop, right? But over here it's opposite. Here you have GC-rich organism with fewer A's and T's, and that means stop codons don't appear randomly as often anymore. And you can travel much further along the genome without seeing stops. So that's the explanation for the black curves here being different. The gray bars are the actual the actual length of the ORFs in these genomes. And even if both pictures show the same, it's difficult to see here because the values are so small. Let's focus on this one instead. And what we see here is that when we get to the longer lengths, there are relatively many more of them in reality than it should have been if this was purely statistically random, right? That's the black curve. But the gray is what we see in reality. So it means that in any biological genome, you could say, the long ORFs are overrepresented. There are many more of the long ORFs than there actually should be if it was completely random, which is the black curve. And this is, of course, because during evolution, longer ORFs have been selected because they can code for real proteins, right? You need a certain length of the gene in order to code for a certain protein. And most proteins are, well, biggish, if not very big, at least they are not super small. So that's why we tend to see longer ORFs in real genomes compared to if we just simulated a random genomic sequence and collected the ORF lengths. So this picture here is actually evolution in seen. You can actually see evolution in this picture. Some coding genes are, however, short. There are some actual coding genes down here as well, in a very short, sadly, you could say, because these are horrible to find, right? Because there are so many random ORFs here. So this is really needle in a big haystack. While up here, it's not, right? Up here, it's only needles and almost no hay in the stack, right? So then the needles are easy to find. But down here, you have a lot of hay and very few needles. And this is why ORF length is such an important feature. If you find some really long ORFs, they are almost certainly real genes, because the probability of being that long is kind of small. However, there are some ORFs who are long, we should remember. But and also, I should mention that software like Prodigal that we will use will discard ORFs below 30 codons. So when you come down here, it gives up. Right? It's not because there couldn't be a gene that short. It could very well, and there are some known genes who are actually that short, but the gene finding machine gives up because this is too difficult, right? Finding these very short ORFs among so many other short ORFs. And remember that even out here, there could be some false positives, right? There are some probability that a random ORF can be this long, actually. But the longer, the more likely it's a real gene. Then the ORF content. And this is what the gene finder papers like to talk about, because this is the fun modeling part. And here I present only one way of looking at ORF content, and that's the codon bias. So what is meant by that? Well, we know that most amino acids are coded by several codons, right? So different codons code for the same amino acid. But if you look into one particular genome, it's quite typical that you see that some codons are preferred, are typical, and some are non-typical. They are not preferred. So within one and the same organism, some of these genomes are used each time almost when you code for a certain amino acid. And the other codons that also code for the same amino acid, they are not used. And this is sort of a feature, like a fingerprint that you can start look for. The problem is that it varies a lot between the organisms. So it's difficult to sort of make use of this. If you have discovered this pattern for one organism, you cannot use it on a different organism because the pattern will be different. What we in principle can do is to make some probabilistic models based on this, but the problem is to have the training data, right? We need some genomes where we know the actual genes, and these genomes must be quite similar to the ones we are looking at, the new genomes we are looking at now. So that's the problem. I made an example though here where I used one of these reference genomes that we are going to use in assignment 3. You are going to use this. And then I took all the actual coding genes in this reference genome. This is one of the best annotated genomes we have. And then I made a sort of a model from that information, and I scored all the LORFs in that genome, both those who are actual genes and all the others, rubbish, who are not genes, right? And this is how the scores turn out. So here I have the score along the x-axis here. And then you can see the green curve is now the distribution for the real genes. And they tend to have bigger scores, right? The vast majority of them have positive score and also 5, 6, 7 or something. The actual values here are not so important. Depends on my model, of course. But we can see the red curve is the distribution for the rubbish ORFs, those who are not real genes, and they tend to be lower, right? Of course, there are some overlaps here, right? So it's not the perfect separation between actual genes and rubbish ORFs. There are even some rubbish ORFs who score pretty well, and there are some true genes who scores pretty bad. But at least the separation is distinguishable, and more than that, I would say this is actually quite good. And I have to say that this is also an organism where this type of analysis works very well. It's not that good usually. This is a sort of a best case scenario in a sense. But it tells us that it's possible to some degree to look at the content of the ORFs, and if you know what to look for, you can say, you can actually find the genes to quite a good extent. And notice that this works on all the genes, right? Even if the genes have completely different amino acid composition, right? Because that if you translate the genes into proteins, the proteins will be wildly different from each other. So you cannot make a sort of sequence alignment type of study here, because that will not work. But this codon bias seems to be the same for almost all the genes, right? Regardless of how they look like actually in their sequence. The upstream motif, I'm talking about what we know as the ribosomal binding site here. And we have learned that from basic microbiology that upstream of the genes, there should be some ribosomal binding site. So once the genes have been transcribed, this is how the transcript binds to the ribosome before translation, right? And there should be some sort of pattern here to look for, a motif. And it should be like 5 to 15 bases upstream of the start codon. And it should also be AG rich, at least that's what the textbooks tell us. And this comes from this Shine-Dalgarno sequence in E. coli, right? That's how this statement has arisen. But if you actually go looking for this AGG motif in the upstream regions of E. coli, you will sadly not find it very often. So here I have, this is also one of the reference genomes, E. coli K12, perhaps the most studied organisms on Earth. If you go hunting for the upstream regions of the genes, here we have the genes of that organism, and these are we are pretty sure that these are the actual correct genes now, because this has been studied over years now, right? 4,140 genes in this organism. Only 113 of them has this pattern in the upstream region. Small minority, actually. So you cannot go looking for this and think that this is what help you find all the genes. It will help you find 113 genes, but vast majority will not have this pattern. So this was a bad signal. And here we can see a separation of the genes in E. coli K12 based on this upstream information. And now I didn't really use this pattern. I just collected all the 4,140 genes, the upstream sequence from them, and then I extracted whatever was sort of overrepresented in this upstream region and used this to score all the genes, right? So I have a sort of a motif score here now. And we can see that the actual gene score is a little bit higher than the rubbish, but not by far as good as in the previous picture. There's a much bigger overlap here now. And also what I actually found when I did this was that this AGG pattern is not the most prevalent one. This is not what characterizes the upstream. This is what is the most characteristic formare in the upstream sequence. Right? If we collect bases of length 4, k-mers of length 4, formers, this is the most common one. And only here we find pieces of this one, right? As the second most common one. And then we have TT. So I mean, it's a little bit mystery to me why this pattern here has been described as what you should sort of look for in E. coli in order to find. This is not the case, really. I think it's based on some very old data and hasn't really been updated. Now we know a lot more about this E. coli than we did some years ago. Well, overlap between genes is also an important piece of information because genes are presumed to not overlap very much. The problem is that there is no really good sort of argument for this, I think. And I think that genes can overlap, not always. I can see that some genes cannot. For instance, two genes who must be transcribed more or less at the same time. They cannot overlap because you can only transcribe one of them and the other one is sort of blocked while you transcribe the first one, right? So genes that must be transcribed sort of consecutively or almost at the same time, they must be next to each other, not on top of each other. But genes who are not linked in this way, they can very well overlap, I think. There is no sort of argument against it. And the problem is that this is used as an heuristic in the software tools, and it's not very much mentioned in the papers that describe these software tools. And it turns out it's quite important. And in Prodigal, the software we'll use, they say they have a sort of a very crisp threshold at 60 bases. So if you have 60 bases overlap, it's fine, no problem. But if it's 61, it's not allowed. So there's sort of a sharp limit here. Why should it be like that. I don't believe that this reflects biology at all. There is no sort of sharp threshold at 60. And why should it be 60 bases? It sounds like someone just decided randomly. There is no argument for 60 bases. It could be just 40 or 50 or 33 or whatever. But this is sort of hidden inside the software, right? Be aware of this. Be aware of this. So if we look at the information used to find genes, I would rank them like this. ORF length is the most important piece of information. And that's very easy to collect and understand, right? The longer, the better. Then overlap between the other genes turns out to be pretty important. And then this sort of choices like this becomes very important, right? The ORF content is not that effective, but it has some impact, and this could of course be more effective in some cases and less effective in other cases, depending on the organisms, right? And I would say this upstream motif information is the least important one here. It could be used and help you to decide a few cases perhaps, but basically you need these others first to really find the genes. Let's end this by saying a few words about evaluating gene prediction. So assume that we have a genome where we actually know all the genes. And we don't need to do any prediction, we have them already. But then we pretend that we don't have them, right? And we use the software to predict the genes using the genome and we hide away this information about knowing all the genes. And then once we have the predictions, we compare them to the actual genes. So then we sort of bring this forward again, this information here. And this way we can actually evaluate. And we typically do that by making this confusion matrix, we call it, this 2x2 table, where we list in the columns what we actually know, right? These are all the ORs who are genes and all the ORs who are not genes in this genome, right? And then in the rows, here are all the ORs that were predicted by the software to be genes, and here are the ORs predicted not to be genes. And then inside the table we have numbers. Here we now count, this is a number, we count how many of the actual genes were also predicted to be genes. And these we call true positives, TP. So this is a number here. And the bigger this is, the better, right? Ideally, we would have all the true genes to also be predicted as true genes. But of course, we must expect to have some errors. And here are one way of making an error, the false positives. Those are all the cases where we predict a gene which is actually not a gene. That's a false positive. And the other way of making an error is down here, where the ORF is actually a gene, but it's predicted not to be one. So that's the false negatives. So the errors are in these two cells here. And these numbers should be as close to 0 as possible, right? That would be very nice. If this one is like, let's say the organism has 3,000 genes. If there's 3,000 here, 0 here, and 0 here, it's a perfect result, right? Perfect result. All the genes are predicted and no others, no errors in either way. This never occurs. But how close do we get? How close do we get? Precision is one statistic which we typically compute from a table like this. If we have a table like this with these three numbers here, we don't need the fourth cell. That's the true negatives, but we don't really need that. We need only these three blue cells here, the values in these three here. The precision is the probability that the predicted gene is actually a gene. And here's the formula. And you can see it takes the true positives, this value, and divide by the number of true positives plus false positives, which is the sum here. And you can see that the sum in these two cells is all the predicted ones. So the fraction of true positives among all the predicted ones, that's the precision. The recall or sensitivity is slightly different. Probability that the actual gene is predicted to be a gene. And here is the formula for the recall. And you can see the numerator is the same, but the denominator is different. So now we take this again and divide by the sum in these two cells instead. And that is the number of actual genes, right? The sum in these two cells. And then the fraction of that is the recall or sensitivity. And both of them should be as large as possible. You can see that these will always be numbers between 0 and 1, both of them, because of these fractions. So if you think of the false positive is 0, then you can see this is 1, right? That's the best you can have. And also down here, if the false negatives are 0, this is also 1. That's the best you can have here. So both of them should be as big as possible, which means as close to 1 as possible. And if they are close to 0, it's horrible. Because then it means this one is small, right? And these two are big. So this is how we can evaluate gene prediction, and you will do this in assignment 3 actually. Gene prediction tools, well, we will look at Prodigal in this course. Glimmer is also a popular tool. GeneMark-S, the Quast software we used can actually use GeneMark-S to predict genes from our contigs and then say something about the contigs based on that. EasyGene is another tool. The point is all of them make mistakes, right? And quite many actually, not percentage necessarily as a big percentage of errors, but remember that we predict many genes, right? In a genome there are thousands of genes, and if you have like 10% error, 10% of thousands is still a big number, right? So they make plenty of mistakes. They also tend to be what we call oversensitive, and that's probably by design, I would say. So they have a tendency of producing many false positives because they want to have all the true, as large number of true positives as possible. So they tolerate also quite a big number of false positives, not as many false negatives. Missing a gene is considered worse than having some extra rubbish in there. That seems to be the order. And there's some good reasons for this. For instance, if you go looking for gene expression, you want to see how the genes are expressed. Then missing a gene can be catastrophic. But it doesn't matter if you have a lot of rubbish genes on your list because they will never be expressed anyway, right? So when you start measuring gene expression, you will see that they have 0 expression in all cases, and that's not a problem then. So I think that this is the reason for this oversensitivity. These tools have been designed to be oversensitive because it's a good idea for looking for gene expression. But in some other cases, it's a bad idea. For instance, we will later in this course talk about pangenome analysis and core genes and cloud genes. And especially these cloud genes, those are genes who appear rarely in different genomes. They are typically suffering a lot. I mean, we have all the false positives tend to turn up as cloud genes. So this will affect that type of analysis rather heavily. So for some cases, oversensitivity is fine, but for others, it's horrible. And I should also say that because we have this oversensitivity, the annotation becomes important. So assigning some function to these predicted genes, that's the next step. And we will do that, but not in this talk. And typically those predicted genes that gets no annotation, they are candidates for being the false positives, right? So we can actually prune the list of predicted genes after the annotation. I think that's quite common to do. You predict genes, get a lot of false positives, and then you annotate and see which of these did not get an annotation. Okay, let's put them aside as false positives, or at least be skeptical of these genes, predicted genes, right? Because they are most likely errors. So let's see how we can find a Singularity container for InterProScan and install this on Orion. So first of all, I tried to look up in the ordinary channels, you could say, see my text on module 6, where you search for containers, but I didn't really find a Singularity container for InterProScan on those websites, you could say. So what I do then, I go Googling, of course. InterProScan. And I want to search for Singularity container. So this is my search phrase, right? Software I want to look for, and also that I want a Singularity container. Let's see what happens. And actually the very first hit here is the one I ended up with at a place called Agbase. Let's have a look at this site. Make it a little bit bigger here so you can see. So this is actually a very nice site, I would say, because they explain very carefully how to proceed to make this work. That's not really always the case. First, it's about downloading the InterProScan database, right? Which is a very huge one, and we don't want to do that. Here you can see how to do it if you are on Orion. Use the vget and then the FTP address to where you find this data. But these are you don't do this. These are many gigabytes. I have done it and it's installed on Orion, so we have everything we need. See my module 6 document. There's a mentioning of it there. What I want to do is to scroll down to container technologies. So here it says how to run InterProScan using Docker. And Docker is a software very much similar to Singularity that we have used. On Orion, we cannot run Docker, but we can run something called uDocker. But we will not do that because we stick to a Singularity that we have used before. Here, however, is the command for pulling the Docker image, as we call the container. So this one I just copy now, and we will see later why I copy this one. If we scroll further down here, we will also see that there is a section about how to use Singularity. Here it comes. And this is not always the case, I have to say, because this is actually a Docker image, and usually the people supplying the Docker images, they describe how to do this with Docker and not with Singularity. But these people have actually made the trouble to do that, and that's luxury, I would say. So if we scroll a little bit down, first how to download the data again. This is exactly the same, of course. But then how to get the container. And here is the command for getting actually the same container using Singularity. And notice it's almost the same. And the reason normally I would copy this one, right? Because I want to do the Singularity. Let me now go back to my terminal window with the first address I copied. And now I will show you why I did that. Because first of all, here I am in now my module 6, and I have nothing in my module 6 folder. This is where I want my container to be, right? Module 6. You can put it anywhere, but this is where I'm going to use it later, so let me put it in here. Now I right-click to just paste in what I copied from that website. And this I cannot run because I cannot run Docker. So I have to modify this a little bit. I want to run Singularity like this, yes. The pull is the same. Docker pull or Singularity pull, both do the same thing. So this I don't need to change. And actually the name of the container, or the address, I mean, the only thing I need to change here is to add docker : / before the address. And that's because when you use Docker to pull, this is sort of implicitly understood. But when you use Singularity to pull it, you need to add this to get the full web address. It's on a Docker. It's a Docker address. So this should now work, right? So if there hadn't been any sort of on that website, if they had no explanation of how to do this in Singularity, this is how you could do it. If they just talked about their Docker image, you can pull the Docker image by using Singularity with these small changes, right? Use Singularity instead of Docker, and then add this here to the Docker address. Let's try to pull it now. And then some downloading starts and building of this Singularity image file. So there is some downloading first, and this of course will take some time depending on the size of the container. Some are quick and some takes quite some time. And there will always be some messages and there can be some warnings and stuff, we will see. And lots of warnings here. But as long as there is no error, I will hope this works fine. As long as it's working, I'm happy. And now the pulling is complete without any error messages. And the last statement here is creating the CIF file. CIF is short for Singularity Image File. And if I now make a LSL here, we will see that one file has popped up called interproscan 5.45-80 blah 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 .cif, right? .cif, that's the extension. So this should now be the Singularity Docker, and we can see the size of it here. We can actually remember if we add an H here, you will get it in a more readable, human readable, 380 megabyte. It's not that big, right? 380 megabyte container. And now we should be able to run this container. And we have previously run containers like this, right? And then interproscan. And now we have the container locally here. We don't need this CVMF / Galaxy server, right? Because it's here. We don't need that anymore now because we actually have the container on our own folder. So we can write this straightforward. And then we need some command here in order to run this. And here, this is different for this container because interproscan is started in a very strange way. There is no sort of command to start interproscan. Actually, if you look at the help file for interproscan, if you just Google that, you will find that it's something like this. It's actually a shell script that is just invoked. And this will not work here now because the people that made this container, I tried, so I know. The people that made this container, they have changed this. So actually, I don't know what is the command line command to start, but we don't really need it because instead of exec, we can use run here instead. And then it will automatically look inside the container for the command that runs this container, you could say. So now we can just list some option here, and -h is the first thing I would try without knowing anything about this software. Can I run this with a -h to get some help text, perhaps? Let's try. Yes, amazingly, it worked. So I have actually now downloaded and sort of installed interproscan locally here, and it seems to work by this run statement. So this you should also do, and then you can go back to my Module 6 document to see how we will actually run into Proscan with some more options and stuff here. We will bind in some local folders and stuff, so it's actually sort of complicated compared to the other containers we have used. But it's nice to have seen this at least once. But anyway, it was really easy to download or pull this container and make it work.